Chernobyl nematodes appear to tolerate high levels of radiation. Scientists studying nematodes living in the exclusion zone around the Chernobyl nuclear power plant have shown that these creatures appear to be resistant to the effects of the 1986 disaster. Constant exposure to high levels of ionizing radiation did not damage their genomes. In late April 1986, after an unexpected loss of power during a safety test, the Unit 4 reactor at the Chernobyl nuclear power plant melted down. The steam from the explosion released a wave of radioactive material that spread across Europe, contributing to health problems for thousands of people. The disaster at the Chernobyl nuclear power plant transformed the area. People were evacuated, but many plants and animals still live in the region, despite high levels of radiation that persist almost 4 decades after the accident. But, as New York scientists have found, constant exposure to such high levels of ionizing radiation does not seem to have any significant impact on the microscopic nematodes living in the Chernobyl zone, suggesting that they are exceptionally resistant to such threats. The results and description of research on Chernobyl nematodes were published in the journal, Proceedings of the National Academy of Sciences. The exclusion zone around the Chernobyl nuclear power plant covers 2,600 square kilometers in the Kiev and Zhytomyr oblasts. In recent years, scientists have discovered that some of the animals living there are physically and genetically different from their counterparts in other parts of the country, raising questions about the effects of radiation on DNA. In a study published last month, scientists found that wolves living in the zone are different from their cousins outside the zone, and that these changes mainly affect their immune systems and appear to increase the wolves' resistance to cancer. Previous research on the genomes of feral dogs living in the exclusion zone showed that there are significant genetic differences between two groups of dogs living in different areas of the zone. In turn, in analyses from 2022, scientists discovered a probable protective mechanism against ionizing radiation that evolved among frogs near the power plant. In a new study, scientists from New York University looked at nematodes. They showed that chronic exposure to high levels of ionizing radiation did not damage their genomes. Nematodes are tiny creatures with relatively simple genomes. They reproduce quickly, and this, combined with the structure of their genome, makes them excellent organisms for studying basic biological phenomena. Nematodes live everywhere and they live fast. So they go through dozens of generations of evolution while a typical vertebrate is still putting on its shoes, said Matthew Rockman, a professor of biology at New York University and an author of the study. I saw footage of the exclusion zone and was surprised how lush and overgrown it was. I never thought of it as a bustling zone. When looking for organisms that are particularly tolerant to radiation, this is a good place, admitted Sophia Tintori from New York University, co-author of the publication. In cooperation with biologist Timothy Musso from the University of South Carolina and scientists from Ukraine, scientists visited the Chernobyl zone in 2019. Armed with Geiger counters to measure radiation levels and protective equipment, Scientists collected samples of soil, rotting fruit and other organic material. Samples were collected from locations throughout the zone with varying radiation intensities. The samples were subjected to preliminary analysis in a laboratory set up in one of the abandoned apartments near the power plant. Scientists found hundreds of nematodes in them, which were isolated from samples and secured during the trip to the USA. In a laboratory at New York University, scientists continued to study nematodes. We can freeze them and then defrost them for later research. This means we can stop evolution in the lab, which is impossible in most other animal models and very valuable when we want to compare animals that have experienced different evolutionary histories, Rockman said. In their analyses, the scientists focused on nematodes of the species Oshia stipuli. They sequenced the genomes of 15 creatures from Chernobyl and compared them with the genomes of 5 nematodes of the same species from other parts of the world. Interestingly, scientists, despite using various methods, failed to find any signs of genome damage caused by high levels of radiation. This does not mean that Chernobyl is safe. Rather, it means that nematodes are truly hardy animals and can withstand extreme conditions. We don't know how long each of the individuals we found was in the zone, so we can't be sure what levels of radiation it and its ancestors were exposed to over the last 40 years, Tintori noted. The research team wondered whether the lack of visible damage to the genome was due to their resistance to radiation or perhaps to their effectiveness in repairing the damage. In the next step, the researchers developed a system that compared the growth rate of nematode populations and used it to measure the sensitivity of the descendants of each of 20 genetically distinct nematodes to different types of DNA damage. Although the different nematode lines differed from each other in their tolerance of DNA damage, these differences did not correspond to the radiation levels from the locations where they were collected. This suggests that the Chernobyl nematodes are not necessarily more tolerant to radiation, and that radiation did not force them to evolve. The results provide scientists with clues about how DNA repair may vary from one individual to another. And despite the genetic simplicity of Oshia stipuli, may lead to a better understanding of natural variation in humans. Now that we know which strains of Oshia stipuli are more sensitive and tolerant to DNA damage, we can use them to investigate why different people are more exposed to carcinogens than others, Tintori said. How different individuals within a species respond to DNA damage is a major topic of interest for cancer scientists who want to understand why some people with a genetic predisposition to cancer develop the disease and others do not. Researching how individuals respond differently to DNA-damaging agents in the environment will help us gain a clearer picture of their own risk factors, Tintori added. Recording of entry into the Earth's atmosphere. The W1 capsule recorded the return journey to Earth. The W1 mission capsule from Varda Space Industries landed on Earth on February 21 after many months spent in space. A camera mounted on the capsule recorded its return to Earth in incredible detail. It captured the entire atmospheric entry sequence, from low Earth orbit through landing. The W1 capsule was docked to the Rocket Lab Photon orbital platform for 8 months. Using the camera mounted on board, she documented her entire return journey, from separation from her home in low Earth orbit to the dynamic entry into the atmosphere, followed by parachute opening and landing at the Utah Test and Training Range. A test capsule used to produce drugs in microgravity landed on Earth on February 21. She was carrying a batch of antiviral drugs grown in low Earth orbit, LEO. According to the company's representatives, the speed during the return journey to Earth reached a maximum of Mach 25, which is 25 times faster than the speed of sound. The recording begins by disconnecting from the docking station. After a while, the Earth comes into view. But the real fireworks start about halfway through the recording, when the capsule begins to enter the atmosphere. The full, unedited, according to Varda Space Industries representatives, footage below lasts over 27 minutes in total. Data from the flight and atmospheric entry will be shared with NASA and the U.S. Air Force. Varda Space Industries launched its W1 capsule into space in June 2023. The company's vision is to develop pharmaceutical products in space and send them to Earth via specially developed return capsules. As admitted by the company's representatives, microgravity can improve the quality of manufactured pharmaceuticals. It radically changes buoyancy forces, natural convection and sedimentation. This offers the potential to produce high-quality drugs with improved crystal structures due to the absence of gravitational stress, leading to increased shelf life and improved efficacy. We produce medicines in space. Removing gravity allows us to produce drugs that would not be possible on Earth. Gravity is something like a parameter. If you put the temperature knob on your oven, there's a whole world of new recipes and new dishes you can create. Similarly, if you can change the force of gravity, you can also change the chemical process when creating drugs, said Will Brewy, head of Varda Space Industries, last year. The W1 capsule spent 8 months integrated with the Rocket Lab Photon probe, the upper stage of the Electron rocket, which provided power, communications and control of the capsule's orientation in orbit. The actual production of the drug took much less time. But the company had problems obtaining permission to re-enter the Earth's atmosphere. The return to Earth was originally scheduled for September 2023, but the U.S. Federal Aviation Administration did not grant permission due to safety concerns. Permission was finally granted and a capsule containing an antiviral drug known as ritonavir, used to treat HIV and hepatitis C, reached Earth. Now the pharmaceutical from space will be subjected to detailed tests. The company is also preparing for its second launch. which is planned for this summer. This mission was a phenomenal feat and an impressive display of teamwork between the Rocket Lab and Varda teams to create a unique spacecraft with enormous capabilities. Successfully demonstrate in space manufacturing, and return the capsule and finished pharmaceutical product, all on the first try, he said. Peter Beck, head of Rocket Lab. A small but surprisingly loud fish. Dananella cerebrum fish reach just over a centimeter in length, but their small size does not prevent them from being very noisy. These fish can produce sounds of over 140 decibels, which can be compared to an ambulance siren. Fish are considered rather quiet creatures. However, there are certain species that can be surprisingly noisy. In new research published in the journal PNAS, scientists described a species of small fish that, using a special camera, can generate sounds with an intensity of over 140 decibels. These tiny fish probably use sounds to communicate with each other in murky waters. Fish of the species Dananella cerebrum reach a length of only about 12 millimeters. They are almost transparent and live in the shallow and turbid streams of Burma. This species is also known for having an extremely small brain, 0.6 cubic millimeters, the smallest among vertebrates. This tiny fish can produce sounds with an intensity of over 140 decibels at a distance of 10 to 12 millimeters. This is comparable to the noise a human hears in an airplane during takeoff from a distance of 100 meters, and quite unusual for an animal of such a small size. We tried to understand what mechanisms are responsible for this, explains Dr. Ralph Britz from Senckenberg Natural History Collections in Dresden. The most common mechanism for sound production in fish is the vibration of their swim bladder, a gas-filled organ used to control buoyancy, driven by rhythmic contractions of specialized, drumming, muscles. A team of researchers led by scientists from Charité University in Berlin discovered that the fish has a unique sound production system. Using a combination of high-speed video, microcomputed tomography, and gene expression analysis, the researchers showed that male Dananella cerebrum species have a specific sound-generating apparatus that includes drumming cartilage, specialized ribs, and fatigue-resistant muscle. This allows you to generate fast, loud pulses. The rib next to the swim bladder is moved by a special muscle. This taut piece of cartilage, when released, hits the swim bladder and produces a drumming sound. In males, the rib is much harder, which explains why females of this species do not produce sounds. But why do small fish need such a mechanism? We assume that competition between males in this visually restrictive environment contributed to the development of a special acoustic communication mechanism, says Britz. This mechanism may also help navigate through murky waters, or be an aggressive tactic used by males to warn against competition.